Hello students, welcome to SK Biology. This video is about post-transcriptional modification, that is modification of newly synthesized RNA molecule. This happens mainly in eukaryotes. Let us begin. Post-transcriptional modifications are chemical and structural changes that occurs in a newly synthesized RNA molecule after transcription and before it become a functional and mature RNA. So these modifications are basically for to convert naive RNA molecule into functional and mature RNA. These modifications especially important in eukaryotes, where the primary transcripts like pre-mRNA, hnRNA, they must be processed to form mature mRNA. These modifications increases RNA stability, they carry out nuclear export, and they also ensures accurate translation. In eukaryotes, majorly three types of post-transcriptional modifications are present. The first one is for modification of 5' end, which is 5' capping. The second modification is for modification of 3' end, which is 3' polyadenylation. And the third post-transcriptional modification is to modify intermediate region of RNA, which is RNA splicing. We will discuss all these three post-transcriptional modifications in detail one by one. Let us begin with 5' capping. It is also called as G-capping, as modified 7-methyl guanine nucleotide is added at the 5' end of RNA. Addition is by specific type of bond called as 5' triphosphate linkage. The function of 5' capping is 5' capping protects mRNA from degradation. This modification also helps in ribosome binding during translation, that is protein synthesis. This 5' capping also facilitates nuclear export. Now let us understand how this 5' capping is done on mRNA. So to add G-cap to the mRNA, cell needs 3 different enzymes for 3 different steps. The first step is done by enzyme RNA triphosphatase. It removes one phosphate from 5' end of new RNA synthesized and convert triphosphate nucleotide to diphosphate nucleotide. Once diphosphate is formed, second enzyme, that is guanylyl transferase, comes in function. It adds GMP, that is guanine monophosphate, from GTP to 5' end of RNA via 5' triphosphate linkage. After addition of guanine molecule, the third step is methylation, which is done by third enzyme methyl transferase. This enzyme methylates guanine at N7 position and forms N7 methyl guanosine cap, that is M7G. Methyl group he get from donor called as SAM, that is S-adenosine methionine. So action of these three enzymes add G-cap to the 5' end of newly synthesized RNA molecule. So this is all about post-transcriptional modification 5' capping. Next modification we will discuss in next lecture.